Here is a question that should unsettle you. Life existed on Earth for 3 billion years, and almost nothing happened. 3 billion years of bacteria, of algae, of simple, single-celled organisms drifting in the ocean and building stromatolite mats on the seafloor. 3 billion years without a single eye, without a single tooth, without a single pair of legs, without anything you would recognize as an animal in any conventional sense. And then, in a window of time so narrow that geologists call it an instant, 20 million years out of 3.8 billion, evolution invented everything. Every basic body plan that has ever existed on Earth appeared in that single brief interval. Not some of them, all of them. The plan that produces fish, and the plan that produces insects, and the plan that produces worms, and the plan that produces you. All of it appeared in 20 million years after 3 billion years of biological quiet. Why? That question has haunted evolutionary biology since Darwin himself admitted it was one of the most serious objections to his theory. And as of April 2026, we are closer to an answer than we have ever been. But we are still not all the way there. This is the story of the most creative 20 million years in the history of life on Earth. Before we go any further, please subscribe to this channel. Your support really helps us to continue this work. Thank you. There is a number that puts everything else in perspective. 3 billion years. That is how long life existed on Earth before the Cambrian explosion. To put it another way, if you compress the entire history of life on this planet into a single calendar year, with the first living cells appearing on January 1st, then complex animals do not appear until approximately the 20th of November. And the Cambrian explosion, the 20 million year interval in which virtually all animal body plans appeared, occupies about 4 days of that compressed year. 4 days out of 365. In those 4 days, the ocean went from being populated primarily by soft, immobile organisms feeding on microbial mats to being populated by animals with eyes, claws, shells, guts, muscles, and brains. Animals that hunted and fled, burrowed and swam, filtered and crushed. Animals from which every creature alive today is descended. The question of why this happened when it did, why 3 billion years of biological quiet was followed by 20 million years of explosive creativity, is one of the deepest unsolved problems in all of science. It is not merely a paleontological curiosity. It touches on the fundamental question of whether complex animal life was inevitable, given sufficient time, or whether it required a specific and improbable combination of circumstances that may be rare in the universe. The answer to the Cambrian explosion is, in a very real sense, the answer to the question of how likely it is that complex life exists elsewhere. Before we can understand the explosion, we need to understand what came before it. And what came before it was, by any measure of biological complexity, almost nothing. 600 million years ago, the Earth was emerging from the most extreme climate event in the planet's history. The Snowball Earth glaciations, the Sturtian, beginning approximately 717 million years ago, and the Marinoan, ending approximately 635 million years ago, had covered the planet in ice from pole to equator. The ocean, under its ice, had been effectively sealed from the atmosphere for tens of millions of years. Photosynthesis had been reduced to a thin zone near the ice surface in the tropics. Life had retreated to the hydrothermal vents of the ocean floor and the few ice-free refugia that remained. And then, when the ice finally melted, driven by the slow accumulation of volcanic carbon dioxide that eventually overwhelmed the ice albedo feedback, the ocean that emerged was chemically extraordinary. The post-snowball ocean was rich in dissolved minerals that had accumulated over millions of years of volcanic input without biological consumption. It was rich in calcium. It was rich in phosphate. It was, in a very specific chemical sense, ready for biology to do something new. And the first thing biology did was produce the Ediacaran fauna. The Ediacaran organisms, which appear in the fossil record beginning approximately 575 million years ago and persist until approximately 539 million years ago, are among the most beautiful and most puzzling fossils in the geological record. They are preserved as impressions in fine-grained sandstone, and they look like nothing alive today. Dickinsonia, an oval, ribbed organism that grew to lengths of over 1 meter, about 3 feet, moved across the seafloor by absorbing nutrients through its body surface, leaving feeding traces that are preserved alongside its body impressions. Charnia was a frond-like organism anchored to the seafloor and extending upward into the water column, filter feeding on organic particles. Cambrian, arguably the most animal-like of the Ediacaran organisms, with evidence of a mouth, a muscular foot for locomotion, and feeding traces made by a rasping structure, may be an early relative of the mollusks. For decades, the Ediacaran fauna was interpreted as a group of entirely enigmatic organisms with no relationship to any modern animal phylum, a failed evolutionary experiment that went extinct at the end of the Ediacaran and left no descendants. The more recent view, which has gained significant ground in the past decade, is that the Ediacarans were a mixed group. Some were genuine animals related to modern phyla, others were algae or fungus-like organisms, and a few may indeed represent body plans with no modern descendants. The question of what the Ediacaran fauna actually were remains one of the most actively debated in all of paleontology. What is not debated is the nature of the world they inhabited. The Ediacaran Ocean was a world without predators in any meaningful sense. There were no animals with teeth, no animals with claws, no animals that actively pursued and captured prey. The ecological interactions that dominated this world were simple. Organisms fed on the microbial mats that covered the seafloor, on dissolved organic matter in the water column, or on the bodies of other organisms that had already died. There was no need for armor because nothing was trying to eat you. There was no need for eyes because nothing was trying to be seen. There was no need for speed because nothing required escape. The Ediacaran world was, in the most literal ecological sense, peaceful, and that peace was about to be shattered. But before we get to the explosion itself, one of the most significant recent discoveries in paleontology has fundamentally revised our understanding of when exactly the biological revolution began. In April of 2026, a team of researchers studying the Jiangchuan fossil site in southwest China published a paper describing an ecosystem of remarkable complexity dated to between 554 and 539 million years ago, the late Ediacaran, before the conventional start of the Cambrian period. Among the fossils found at Jiangchuan were early relatives of starfish, worm-like creatures with bilateral symmetry, and organisms that show features consistent with early deuterostomies, the broad group that includes all vertebrates, including humans. This discovery is significant in multiple directions simultaneously. First, it suggests that the roots of the Cambrian explosion were already visible in the late Ediacaran, that the diversification of major animal groups began earlier than the start of the Cambrian period itself. Second, it supports the hypothesis that the Cambrian boundary, as defined in the geological record, represents not the beginning of animal evolution, but rather the beginning of the interval in which animals started producing hard parts, shells and skeletons that preserve readily in the fossil record. The animals may have been there before the shells. We just could not see them. This idea was reinforced by another major finding from June of 2025. A team from the University of Barcelona, analyzing 545-million-year-old fossil trails from the late Ediacaran of Spain, argued that the locomotion traces show the scaling laws characteristic of bilaterian animals. Animals with a definite front and back, top and bottom, moving purposefully through the sediment. If their interpretation is correct, these traces represent the earliest known evidence of bilaterian locomotion, predating the conventional start of the Cambrian by roughly 6 million years. The Cambrian explosion, it appears, had a fuse that had been burning before the Cambrian began. The Ediacaran organisms deserve more than a brief catalogue because they represent the last chapter of a world that was about to end. And understanding what was lost helps us understand what the Cambrian explosion actually created. Dickinsonia, the oval-ribbed organism that grew to over a meter in length, is now understood through remarkable isotopic evidence published in 2018 to have been an animal rather than a fungus or alga. The cholesterol preserved in its body impressions is distinctively animal in character, making Dickinsonia the oldest known animal with confident chemical confirmation. But Dickinsonia was not an animal in any sense that would be familiar today. It had no mouth, no gut, no nervous system that we can detect. It absorbed nutrients directly through its ventral surface in contact with the microbial mat below, essentially digesting the mat from above. It grew by adding new segments at the rear, while older segments grew proportionally, a growth pattern unlike anything in modern zoology. Cambrian, the most behaviorally sophisticated of the Ediacaran organisms, left behind feeding traces in the form of fan-shaped scratches in the sediment ahead of its direction of travel, made by a rasping structure analogous to the radula of modern mollusks. These traces, found at multiple Ediacaran sites in Russia and Australia, represent the oldest known evidence of directed locomotion and active feeding behavior in the fossil record. Kimberella was going somewhere. It was looking for something. It was, in its own way, the first animal to have a behavioral life that we can reconstruct from the fossil record. The White Sea region of Russia and the Ediacaran hills of South Australia, the two richest Ediacaran fossil sites in the world, preserve these organisms in fine-grained sandstone through a mechanism that was only recently explained. A January 2026 study published in the journal Gondwana Research found that Ediacaran preservation was enabled by the unusual seawater chemistry of the late Proterozoic ocean, which created clay cements around the bodies of organisms after burial, a kind of natural casting process that captured delicate shapes that would normally vanish within days of death under normal burial conditions. The specific chemistry of the Ediacaran ocean was, in this sense, both the environment in which these organisms evolved and the medium that preserved them for us to find 570 million years later. What was completely absent from the Ediacaran world, and this absence is the most important fact about it, was bioturbation. The sea floor of the Ediacaran was covered by a continuous, unbroken microbial mat. This mat was not merely a background feature of the environment. It was the dominant ecological entity of the Ediacaran world, covering virtually every square centimeter of seafloor in the shallow marine environment and representing the primary food source for most of the organisms living there. And it was perfectly intact because nothing was digging through it. No worms burrowing through the sediment, no crustaceans stirring the bottom, no fish rooting through the substrate. The Ediacaran seafloor was, in geological terminology, a mat ground, and it would remain a mat ground until the earliest Cambrian, when the first burrowing animals began the process of destroying it. The chemical conditions that enabled this fuse to ignite have been studied intensively for decades, and the most recent synthesis points to the oxygenation of the deep ocean as one of the critical enabling factors. For most of Earth's history, the deep ocean was anoxic, devoid of free oxygen. The surface ocean, where photosynthetic organisms lived, had been oxygenating gradually since the Great Oxidation Event 2.4 billion years ago. But the oxygenation of the deep ocean, the abyss where most of Earth's water volume resides, lagged far behind the surface. The deep ocean remained a chemically reducing, sulfide-rich environment that could not support aerobic metabolism in complex animals for hundreds of millions of years after oxygen first appeared in the atmosphere. The progressive oxygenation of the deep ocean through the late Proterozoic, and especially through the Ediacaran and earliest Cambrian, was not simply a permissive factor. It was an active driver. Complex animals with high metabolic rates require substantial quantities of dissolved oxygen to fuel their tissues. The evolution of eyes, muscles, guts, nervous systems, and the active locomotion that characterizes the Cambrian fauna all require energy budgets far exceeding anything available to the simple organisms of the Proterozoic ocean. Without dissolved oxygen, there is no energetic substrate for biological complexity. As the deep ocean oxygenated through the Ediacaran, the energetic threshold for complex animal evolution was crossed. But oxygen was not sufficient. If oxygen alone were the answer, Complex animals would have appeared progressively as oxygen levels rose, and there would be no explosion, just a gradual increasing complexity over millions of years. The explosion requires an additional explanation, and that explanation has become increasingly clear: predation. 539 million years ago, at the very base of the Cambrian period, something unprecedented appeared in the fossil record: minerals, biological minerals, tiny shells and tubes, and plates of calcium carbonate and calcium phosphate, produced by organisms that had previously been entirely soft-bodied. This event, the simultaneous appearance of mineralized hard parts in multiple unrelated lineages at essentially the same geological moment, is called the biomineralization event, and it is one of the most striking features of the Cambrian boundary. The small shelly fauna, the assemblage of tiny, isolated mineralized pieces that appears globally at the very base of the Cambrian in rocks worldwide, represents the first wave of this revolution. Before the first trilobites scuttled across the Cambrian seafloor, before Anomalocaris patrolled the Cambrian waters, the ocean was already filling with the disarticulated remains of organisms whose external skeletons had broken up into thousands of tiny plates and spines and tubes. Reconstructing the original animals from these fragments has been one of the great challenges of Cambrian paleontology. Many of the small shelly organisms, Halkieria, Microdictyon, Wiwaxia, turned out, when articulated specimens were eventually found, to be parts of the same animal, a single creature covered in hundreds of interlocking sclerites or armor plates that scattered after death. Why did mineralization appear simultaneously in so many unrelated lineages at the very beginning of the Cambrian? This is the question that cuts to the heart of the explosion. The materials needed, calcium, carbonate, phosphate, had been available in the ocean for billions of years before the Cambrian. The biochemical pathways for precipitating these minerals are not trivially simple, but neither are they extraordinarily complex. The simultaneous appearance of biomineralization in dozens of lineages cannot be explained by independent evolutionary invention at the same time. It must be explained by a shared external pressure, a selective force that acted simultaneously on all of them. That force was predation. The simultaneous appearance of mineralized armor across unrelated lineages is exactly what you would expect if a predator had appeared and began hunting soft-bodied organisms across the entire ocean simultaneously. The shell was not an invention, it was an answer, and answers only appear when there is a question, in this case, something in the water that was trying to eat you. In November of 2025, a research team from Virginia Tech published a paper in Science Daily describing a 540 million year old fossil that they argued was rewriting our understanding of the biomineralization event. The organism in question, Saltarella, a peculiar conical shell fossil that had puzzled paleontologists for decades, turned out to belong to the Cnidarian family, the group that includes modern corals, jellyfish, and sea anemones. Saltarella was remarkable because it combined two different mineral building methods simultaneously: a strategy that had never been seen before in any organism. The finding, the researchers argued, illustrates how the biomineralization event was not simply a single innovation that spread across the animal kingdom, but rather multiple independent solutions to the same problem: how to build a hard protective structure in an ocean that had suddenly become dangerous. The evidence for predation as the primary driver of the Cambrian explosion has been accumulating for decades. But a remarkable new theoretical synthesis published in the journal Frontiers in Ecology and Evolution in January of 2026 has pushed this argument to a new level. The paper, by a team from the University of Nebraska, proposes that the ultimate trigger for the explosion was cellular predation among the single-celled ancestors of animals, predator-prey interactions at the microscopic level that created the genomic instability needed for rapid architectural innovation. Their argument, which we will examine in detail in a later act, connects the macroscopic arms race visible in the fossil record to the molecular mechanisms of genomic change that produced the body plan diversity of the Cambrian. Before we get there, we need to spend time with the fossils themselves. Because the Cambrian explosion is not merely a theoretical problem in evolutionary biology, it is one of the most spectacular chapters in the history of life, preserved in extraordinary detail in a series of fossil sites that have transformed our understanding of what the Cambrian world actually looked like. The Burgess Shale of British Columbia is the most celebrated Cambrian fossil site in the world, and its discovery story has the quality of myth. In August of 1909, the American paleontologist Charles Walcott was riding along a trail in the Canadian Rockies near the town of Field when his horse stumbled on a loose slab of shale. Walcott dismounted to examine the rock and found, preserved in extraordinary detail in the fine-grained black shale, the soft tissues of animals that had died 508 million years ago. He had discovered the Burgess Shale, a deposit of such extraordinary preservation quality that it would transform paleontology and provide the primary window into the Cambrian world for the next century. The Burgess Shale preserves what geologists call a Lagerstätte, a German term meaning storage place, a fossil deposit in which exceptional conditions prevented the normal decay of soft tissues before burial, preserving the complete anatomy of the organisms in extraordinary detail. The animals of the Burgess Shale are preserved as carbon films on the shale surface, showing not only their hard parts, exoskeletons, shells, but their muscles, their guts, their eyes, and in some cases even the contents of their digestive tracts. The preservation is so complete that researchers have been able to reconstruct the feeding behavior, locomotion, and ecological relationships of organisms that lived 500 million years ago with a precision that rivals studies of living animals. What the Burgess Shale reveals is a fauna of extraordinary diversity and strangeness. The organisms of the Cambrian Ocean were not primitive or simple in any meaningful sense. They were complex, highly adapted, and in many cases entirely unlike anything alive today. They occupied a full range of ecological niches: predators, scavengers, filter feeders, deposit feeders, and they interacted with each other in an ecological web of considerable sophistication. The Cambrian Ocean was not a simple or primitive place. It was, in its biological richness and ecological complexity, as sophisticated as any marine ecosystem of the modern world. It was just populated by animals so strange that a modern biologist transported back in time would initially struggle to recognize any of them. Anomalocaris, the giant Anomalocaridid, whose name means unusual shrimp, is the most famous predator of the Cambrian seas. Known from the Burgess Shale and from Cambrian deposits worldwide, Anomalocaris reached lengths of approximately 1 meter, about 3 feet, making it by far the largest animal of its time, perhaps 10 times larger than any other Cambrian animal. It was an arthropod relative with 2 large compound eyes on flexible stalks, giving it a wide field of view. 2 segmented, grasping appendages at the front of its head for seizing prey. A circular mouth with radiating plates for crushing hard-shelled organisms. And a series of lateral lobes for propulsion through the water. Its feeding apparatus was powerful enough to crack the exoskeletons of trilobites. The evidence for this is preserved in trilobite fossils with semicircular bite marks matching the diameter of Anomalocaris mouthplates. But Anomalocaris was more than just a large predator. It was the visible expression of the arms race that was driving the entire explosion. The presence of a predator as large and powerful as Anomalocaris in the earliest Cambrian fauna implies that the prey animals it fed on had already evolved defenses sophisticated enough to require a predator of that capability. The shells, the mineralized armor, the burrowing behavior, the camouflage patterns visible in some Burgess Shale organisms, all of these had already been tested and refined by the time Anomalocaris appears in the record. Anomalocaris was not the beginning of the arms race. It was already the middle. Hallucigenia, named for the hallucinatory quality of its appearance when first described, is perhaps the most visually arresting of the Burgess Shale organisms. When first formally described in 1977, it was reconstructed upside down, with what we now know to be its dorsal spines being interpreted as its legs, and what we now know to be its legs being interpreted as dorsal tentacles. The correct orientation was only determined in the 1990s, when specimens from the Chinese Chengjiang deposit showed the complete animal with greater clarity. In its correct reconstruction, Hallucigenia is a small worm-like animal with 7 pairs of walking legs armed with tiny claws, and a corresponding row of paired spines projecting from its back, presumably a defensive adaptation against predators. It belongs to the group of velvet worms, whose living relatives are soft-bodied, caterpillar-like animals found in tropical forests today. Opabenia, with 5 eyes and a flexible, grasping proboscis, was so strange when formally described at a scientific meeting in 1972 that the audience reportedly laughed. The reaction is understandable. An animal with 5 eyes arranged on stalks and a long, flexible trunk ending in a grasping claw is genuinely outside the range of anything in modern zoological experience. Opabenia has since been placed as a relative of the Anomalocaridids, but its 5-eyed face remains one of the most remarkable images in all of paleontology. It is the image that best captures what the Cambrian explosion actually was. Not merely a quantitative increase in the number of species, but a qualitative explosion of biological imagination. An explosion of possible ways to be an animal. Pikaia gracilans, a small blade-like swimming animal preserved in the Burgess Shale, is arguably the most important organism in all of paleontology from a personal perspective, because Picaia, or something very like it, is the ancestor of every vertebrate that has ever lived, including you. Picaia had a notochord, the stiffening rod that runs the length of the body and is the precursor to the vertebral column, and V-shaped muscle blocks on either side for swimming. It was the first chordate, the earliest member of the lineage that would eventually produce fish, amphibians, reptiles, birds, mammals, and humans. At the time of the Burgess Shale, Picaia was not the dominant animal in any ecosystem. It was a small, soft-bodied swimmer, vulnerable to every predator in the Cambrian Sea. Its survival, in the face of Anomalocaris and all the other weapons the Cambrian explosion had produced, was the most consequential act of good fortune in the history of vertebrate life. The Chengjiang biota of Yunnan province in southwestern China is a Burgess Shale equivalent deposit, approximately 10 million years older, dated to approximately 518 million years ago. Discovered in 1984 by the Chinese paleontologist Hao Xiangwang, the Chengjiang fauna has provided an extraordinary complement to the Burgess Shale, not merely duplicating its organisms, but revealing a Cambrian fauna at an earlier stage of its diversification. The Chengjiang fossils preserve organisms with extraordinary three-dimensional fidelity. Not flattened carbon films, but three-dimensional impressions that show the precise geometry of body plans. And they include nervous systems, eyes, and gut contents preserved in unprecedented detail. In August of 2025, a groundbreaking fossil discovery in the Grand Canyon provided a dramatic new window into the Cambrian world. A research team reported the discovery of exquisitely preserved soft-bodied animals from the Cambrian period in the 60-mile formation of the Grand Canyon, a deposit that had never previously been known to contain such preservation quality. The 60-mile formation is one of the oldest Cambrian deposits in North America, and the soft-bodied fauna preserved in it represents one of the earliest snapshots of the Cambrian explosion on the Laurentian continent. The discovery extended the geographic range of exceptional Cambrian preservation to North America at an earlier date than any previously known site, filling a gap in our understanding of how the explosion unfolded geographically. And in January of 2026, a paper published in the journal Nature described perhaps the most remarkable Cambrian fossil site to be discovered in recent years. The Huaiyuan biota of Hunan province in South China preserves a complete Cambrian ecosystem caught in the act of recovery after the Sinsk extinction event, a mass extinction that struck approximately 518 million years ago, devastating Cambrian marine communities worldwide. Nearly 60% of the species found within the Huaiyuan biota are previously unknown to science, suggesting that the recovery from the Sinsk extinction was accompanied by a second, smaller wave of biological innovation. Among the most significant finds in the Huaiyuan biota is the oldest known free-swimming tunicate, a small filter-feeding chordate that provides evidence that modern-style ocean ecosystems were already taking shape in the immediate aftermath of the Cambrian's first mass extinction. The Chengjiang biota deserves its own extended treatment, because in many respects it tells us more about the early Cambrian than the Burgess Shale does. Not because it is more spectacular, but because it is older. 10 million years older than the Burgess Shale, the Chengjiang fauna preserves a Cambrian ecosystem at an earlier and, in some ways, more revealing stage of its development. Among the most significant Chengjiang organisms is Fuxianhuia protensa, a small arthropod whose brain and cardiovascular system are preserved in such detail that researchers have been able to map the structure of what is effectively the oldest brain in the fossil record. The Fuxianhuia brain shows a simplified neural architecture compared to modern arthropods, providing a snapshot of brain evolution at its very earliest stage in the animal kingdom. It is, in the most literal sense, the beginning of the lineage that eventually produced the most complex neural architectures on Earth, including the human brain. The Chengjiang deposit also preserves Unanozoon lividum, a chordate closely related to Picaia, and one of the earliest known members of the lineage that would eventually produce all vertebrates. Like Picaia, Unanozoon was a small, soft-bodied swimmer with a notochord and V-shaped muscle blocks. Specimens from Chengjiang preserve the notochord in sufficient detail to confirm its structure, providing the oldest known physical evidence of the anatomical precursor to the vertebral column. Every spine in your back traces its evolutionary ancestry to the notochord preserved in those Chengjiang fossils from 518 million years ago. Radiodont fossils from Chengjiang include not only Anomalocaris, but also Amplectobelua and several other predatory Anomalocaridids, suggesting that the apex predator guild of the early Cambrian was already diversified into multiple ecological specializations, some pursuing fast-moving prey, others targeting bottom-dwelling organisms, others filtering small particles from the water. The Cambrian arms race had already produced, in its earliest stages, a diversified community of top predators with different hunting strategies. Exactly what you would expect in a mature ecological system, but surprising to find in what was supposedly the dawn of complex animal life. The Wiwaxia corrugata of the Burgess Shale is worth spending additional time with, because it illustrates better than almost any other Cambrian organism how the explosion produced body architectures that have no modern equivalent. Wiwaxia was a flattened oval animal roughly 2 centimeters, about 3/4 of an inch in length, covered on its upper surface with overlapping scales called sclerites, and armed with a row of elongated spines projecting upward from its margin. A defensive palisade that would have made it extremely difficult for a predator to swallow. Its underside was soft and muscular, and it moved across the seafloor by ciliary locomotion. Its feeding apparatus included a radula-like structure for scraping organic material from the microbial mat below. Wiwaxia's systematic position, whether it belongs to the mollusks, the annelid worms, or an entirely separate phylum, has been debated for decades without final resolution. It may represent a body plan that evolved independently of any surviving lineage, and was lost entirely at the end of the Cambrian or Ordovician. The Canadaspis perfecta of the Burgess Shale is, by contrast, easy to place. It is a crustacean, a clear member of the arthropod phylum, and its anatomy is recognizably similar to modern shrimp or krill. Canadaspis is significant not for its strangeness but for its familiarity. It shows that by 508 million years ago, arthropods had already evolved into forms so similar to modern crustaceans that a non-specialist would recognize them immediately. The Cambrian explosion was not only producing strange new body plans, it was also simultaneously producing the first representatives of lineages that have changed relatively little in the subsequent half-billion years. The trilobites deserve particular attention because they are simultaneously the most abundant organisms in the Cambrian fossil record and the most enduring symbol of the explosion itself. Trilobites, named for the three-lobed body plan visible in their fossilized exoskeletons, were marine arthropods with a segmented body protected by a mineralized exoskeleton of calcite that molted as the animal grew, leaving behind a fossil record of extraordinary completeness. They first appear at the very beginning of the Cambrian, 538 million years ago, and they persist until the Permian extinction 252 million years ago, a run of nearly 300 million years that exceeds the current reign of mammals by more than 4 times. The trilobite compound eye is one of the oldest sophisticated visual systems in the fossil record, and some trilobite species evolved eyes of extraordinary optical precision, with individual lenses composed of calcite crystals oriented so as to minimize optical aberration, a solution independently arrived at by human lens makers only in the 17th century. The evolution of image-forming eyes in the earliest Cambrian is, in the view of many paleontologists, the pivotal event that triggered the explosion. Once one animal could see, everything changed. The question of what caused the Cambrian explosion has generated more scientific controversy and produced more competing hypotheses than almost any other question in paleontology. Every decade since Darwin has produced new candidates, new environmental triggers, new genetic mechanisms, new ecological feedbacks that seem to explain the explosion, only to be complicated by new evidence. The current state of the science is not a single answer, but a convergence of multiple factors that together created the conditions for the most creative 20 million years in biological history. The eye is where many researchers begin, and for good reason. The evolution of image-forming eyes, which appear in the trilobites at the very base of the Cambrian, represents a technological threshold whose consequences for the biology of the entire planet were immediate and permanent. Before eyes, the primary sensory modalities of marine animals were chemical and tactile. Organisms could detect the presence of food or danger through chemical gradients or physical contact, but they could not detect objects at a distance. This severely limited both predatory behavior and defensive behavior. You could not chase prey you could not see, and you could not flee from predators you could not detect until they were already upon you. The evolution of eyes changed the rules of every ecological interaction simultaneously. A predator with eyes could detect prey at a distance and pursue them selectively. Prey animals with eyes could detect approaching predators and take evasive action. The entire ecological interaction between predator and prey was transformed from a contact sport into a distance contest, and the distances involved meant that speed, camouflage, armor, and behavioral sophistication all became relevant in ways they had not been before. The arms race that the eye initiated was not merely an increase in the intensity of predation. It was a qualitative transformation of what it meant to be an animal in the ocean. The Cambridge paleontologist Andrew Parker proposed the light switch theory, the hypothesis that the evolution of image-forming eyes was the triggering event for the Cambrian explosion in 2003, and it remains one of the most compelling single-factor explanations for the explosion. Its strength is its specificity. It identifies a single innovation whose consequences are immediate, universal, and exactly proportional to the diversification actually observed. The weakness of the light switch theory is that eyes are themselves complex structures that must have evolved gradually. They do not appear fully formed from nothing, and the process of eye evolution must itself have been enabled by prior conditions. Those prior conditions bring us to the genetic revolution that underlies the explosion. The Hox genes, a family of regulatory genes that control the basic layout of an animal's body, determining where the head goes, where limbs form, and how segments are differentiated, have long been recognized as playing a central role in the Cambrian radiation. Hox genes work as master switches. A single Hox gene can turn on or turn off dozens or hundreds of downstream genes, producing large-scale changes in body architecture through minimal genetic change. The duplication and diversification of Hox gene clusters in the early animal lineage provided the regulatory toolkit from which all the body plans of the Cambrian explosion were constructed. However, as the January 2026 paper in Frontiers in Ecology and Evolution argues with compelling detail, the Hox genes cannot be the primary trigger of the explosion. Molecular clock analyses consistently show that Hox genes diversified before the Cambrian radiation, meaning the genetic toolkit was in place long before the explosion actually occurred. The toolkit was ready. Something else pulled the trigger. That something, according to the Nebraska team, was cellular predation. Their hypothesis proposes that the ultimate driver of the Cambrian explosion was predator-prey interactions among the single-celled ancestors of animals. Not the macroscopic arms race visible in the fossil record, but a microscopic arms race operating at the level of individual cells. When a predatory cell attacks a prey cell and the prey cell escapes, or when a predatory cell suffers damage in the course of a failed predation attempt, the cells involved experience what the researchers call oxidative and enzymatic stress. This stress triggers error-prone DNA repair mechanisms and the activation of transposable elements, genetic parasites that can move around in the genome and insert themselves in new locations. These genomic disruptions, when they occur in developmental genes, can produce chromosomal rearrangements, including Hox cluster reorganization, regulatory network rewiring, and gene duplications with direct consequences for segmentation and body plan architecture. In other words, the microscopic predation happening among the single-celled ancestors of animals was, at the molecular level, generating the genomic variation that eventually produced the body plans of the Cambrian explosion. Predation was not merely the selective pressure that determined which body plans survived. It was also the genomic engine that produced the body plans in the first place. The arms race was not merely external. It was written into the DNA of every cell. This synthesis is compelling because it connects the three levels of the explosion, the environmental conditions, the genetic mechanisms, and the ecological drivers into a single coherent narrative. The rising oxygen of the late Proterozoic provided the energetic substrate for more complex metabolisms. The Hox gene toolkit provided the regulatory architecture for more complex body plans. And the predator-prey arms race, operating simultaneously at the cellular and the organismal level, provided both the genomic variation and the selective pressure that transformed the toolkit into the full diversity of Cambrian body plans. The oxygen story is more complicated and more interesting than the simple version. Rising oxygen enables complex animals, suggests. The geochemical evidence for oxygen levels in the late Proterozoic and earliest Cambrian is derived primarily from isotopic ratios in marine sediments. And the picture that emerges is not of a steady progressive oxygenation, but of a series of pulses and fluctuations, with the deep ocean remaining largely anoxic until a relatively sharp oxygenation event in the latest Ediacaran and earliest Cambrian. Research published by Stocky and colleagues in 2024 and referenced in the 2026 Nebraska paper showed that the oxygen increase was not simply a permissive factor that allowed complex animals to exist. It was an intensity amplifier that increased the encounter rates between predators and prey, accelerated the pace of the predation-induced genomic stress cycle, and supported the higher energetic costs of testing new complex morphologies. The specific number that matters is approximately 6% of modern atmospheric oxygen levels, the threshold below which aerobic metabolism cannot sustain the high-energy muscular activity required for active predation and escape behavior. Below this threshold, animals can exist, but they cannot be fast. They cannot be agile. They cannot engage in the energetically expensive behavioral interactions that drive the arms race. Above this threshold, the arms race becomes possible. The late Proterozoic oxygenation event brought the shallow marine environment above this threshold for the first time, and the consequences were immediate. The phosphate enrichment of the early Cambrian Ocean, another legacy of the Snowball Earth deglaciation, deserves mention as a separate enabling factor. Phosphate is a limiting nutrient in marine ecosystems, controlling the productivity of photosynthetic organisms at the base of the food chain. The unusual phosphate richness of the early Cambrian Ocean, documented in the elevated phosphate concentrations preserved in earliest Cambrian sediments worldwide, supported a level of primary productivity in the shallow ocean that substantially exceeded anything possible in the phosphate-limited Proterozoic ocean. Higher primary productivity meant more food at every trophic level, which meant more energy available for the metabolically expensive process of evolving complex body plans. The Snowball Earth glaciations were, in this sense, not merely a crisis that nearly destroyed life. They were also the geological process that prepared the ocean for the explosion that followed. The bioturbation revolution, the destruction of the Ediacaran mat ground by the first burrowing animals, adds another dimension to this picture, and it deserves its own extended treatment. The calcium chemistry of the early Cambrian ocean also played a direct role in the biomineralization event. The post-Snowball ocean was unusually rich in dissolved calcium, the direct result of millions of years of continental weathering delivering calcium to the ocean without the normal biological consumption that keeps calcium levels in check in modern oceans. This calcium-rich ocean lowered the energetic cost of precipitating calcium carbonate or calcium phosphate into hard structures, making the evolution of mineralized skeletons energetically accessible for organisms that would have found it prohibitively expensive in a calcium-poor ocean. The specific chemical condition of the early Cambrian ocean was, in this sense, a permissive factor for the biomineralization event. The bioturbation revolution, the dramatic transformation of the seafloor from a microbial mat ground to a mixed, bioturbated substrate, is one of the most consequential and least discussed events of the Cambrian explosion. It has its own formal name in stratigraphy, the Cambrian substrate revolution, and it transformed the chemistry, the ecology, and the evolutionary opportunities of the marine environment in ways that amplified and sustained the diversification already underway. Before the Cambrian, the continuous microbial mat covering the seafloor was both an ecological resource and an ecological barrier. As a resource, it provided food for the surface grazing organisms of the Ediacaran. As a barrier, it sealed the sediment below from the overlying water, creating a sharp chemical boundary between the oxygenated water column and the anoxic, sulfide-rich sediment beneath. Nothing burrowed through this mat, because nothing had the muscular apparatus to do so. The mat was effectively impenetrable to biological disruption. The appearance of burrowing animals at the very base of the Cambrian, documented in the trace fossil record by the appearance of simple horizontal burrows in the earliest Cambrian sediments of every continent, shattered this sealed system. As burrowing animals mixed the surface sediment with the overlying oxygenated water, oxygen penetrated downward into the sediment for the first time. This oxygenation of the subsurface sediment created new ecological niches, spaces where organisms could live and feed that had been inaccessible for 3 billion years. It also destroyed the microbial mat that the Ediacaran grazers had depended on, likely contributing to their extinction at the end of the Ediacaran. The engineers of the Cambrian substrate revolution, whatever the first burrowing animals were simultaneously opening new ecological space for themselves and closing the ecological space that supported the organisms they were replacing. The trace fossil record of the Cambrian substrate revolution is the most detailed continuous record of ecological change in the entire Paleozoic. The Treptichnus pedum trace fossil, a distinctive three-dimensional burrow made by an animal moving through the sediment in a specific pattern, is the internationally agreed marker for the base of the Cambrian period itself, precisely because it represents the first evidence of complex burrowing behavior anywhere in the geological record. The appearance of Treptichnus pedum marks not merely the beginning of the Cambrian, but the beginning of the bioturbation revolution, the irreversible transformation of the seafloor from a microbial mat to the mixed, oxygenated, ecologically complex substrate that characterizes all subsequent marine environments. Every animal alive today is a descendant of the Cambrian explosion. This is not a metaphor or an approximation. It is a precise biological statement. Every animal phylum, the highest level taxonomic category above class and order and family that exists today, was established in the Cambrian. Not some of them, all of them. The phylum Chordata, which contains all vertebrates including humans, was established by the Cambrian. The phylum Arthropoda, which contains all insects, crustaceans, and spiders, the most species-rich phylum on Earth, was established in the Cambrian by the trilobites and their relatives. The phylum Mollusca, which contains all snails, clams, and octopuses, was established in the Cambrian. The phylum Echinodermata, which contains all starfish, sea urchins, and sea cucumbers, was established in the Cambrian. Every animal you have ever seen, every animal you have ever read about, every animal that has ever lived, it belongs to a body plan whose fundamental architecture was laid down in those 20 million years. This is the most remarkable fact about the Cambrian explosion, and it is the fact that makes it so difficult to explain. Evolution has continued for 538 million years since the Cambrian, producing hundreds of thousands of new species and extraordinary levels of ecological and behavioral complexity. But it has not produced a new phylum. Not one. In 538 million years of subsequent evolution, no new fundamental body plan has appeared. Everything evolution has done since the Cambrian has been variation and elaboration within the body plans established then. The Cambrian explosion was not merely a rapid period of evolutionary diversification. It was the only period in the history of animal life when the rules of biological architecture were being written from scratch. Why have no new phyla appeared since the Cambrian? This question is as profound as the question of why the explosion happened in the first place. The most compelling answer involves the concept of developmental constraint. Once a body plan becomes established, once the developmental pathways that produce a particular architecture are locked in through millions of years of refinement and the evolution of dependent downstream processes, it becomes essentially impossible to modify the fundamental architecture without destroying the organism entirely. The Hox gene toolkit, once deployed in a particular configuration to produce a particular body plan, cannot be radically reorganized without disrupting every developmental process that depends on it. The first Cambrian animals were, in a sense, writing the rules of animal development as they evolved. By the time the Cambrian was over, the rules were written, and they have not been rewritten since. The organisms that did not survive the Cambrian are in some ways as interesting as those that did. Among the most famous of the Cambrian organisms with no surviving relatives is Opabinia, with its 5 eyes and grasping proboscis. Another is Wiwaxia, a flattened slug-like organism covered in scales and spines that belongs to no modern phylum. Another is the Anomalocaridids themselves, the group that produced Anomalocaris, which went extinct at the end of the Ordovician without leaving descendants. These organisms were not failures. They were successful, widespread, and well adapted to the Cambrian environment. They went extinct not because they were poorly designed, but because the end Ordovician mass extinction, the first great mass extinction in the history of complex animal life, and the subsequent environmental changes eliminated the specific ecological conditions to which they were adapted. Evolution does not preserve the best designs. It preserves the survivors. The question of whether the Cambrian explosion was inevitable, whether 3 billion years of microbial evolution always leads, given sufficient time and the right environmental conditions, to a sudden explosion of complex animal body plans, is one of the deepest questions in astrobiology. If the answer is yes, then complex animal life may be abundant in the universe, because the conditions that enabled the Cambrian explosion, sufficient oxygen, sufficient calcium, sufficient time, and the ecological trigger of predation, are not extraordinarily rare. If the answer is no, if the Cambrian explosion required a specific and improbable combination of geological, chemical, and ecological circumstances that may not be common even on planets with liquid water and microbial life, then complex animal life may be extraordinarily rare in the universe, and our existence becomes more improbable still. The current evidence, taken as a whole, suggests a middle path. The Cambrian explosion required conditions that were necessary, but not sufficient. Rising oxygen, post-snowball chemical richness, the evolution of the Hox toolkit, combined with a trigger, predation, that was not predetermined, but was itself a consequence of prior evolutionary history. The explosion was, in this view, neither inevitable nor miraculous. It was a contingent event that depended on a specific sequence of prior events, but was, given that sequence, ultimately likely. Whether that sequence is common or rare in the universe is a question we do not yet have the observations to answer. Now let me tell you what I think about all of this. The Cambrian explosion is often presented as a mystery, as a puzzle that Darwin identified and that we have been trying to solve ever since. And it is a mystery in the sense that the complete causal chain that connects the chemistry of the late Proterozoic ocean to the full diversity of Cambrian body plans is not yet fully understood. But I think the framing of the explosion as a mystery has sometimes obscured how much we actually know, and how coherent the emerging picture actually is. What we know is this: 3 billion years of microbial evolution produced the chemical conditions, rising oxygen, rich post-snowball ocean, the Hox regulatory toolkit, that made complex animal life energetically and genetically possible. The first animals were soft-bodied, small, and ecologically simple. And then, at some point in the late Ediacaran, something began eating them. The first predator, whatever it was, initiated an arms race that would not stop for 538 million years. The prey animals that survived were the ones that could evade, resist, or defend against being eaten. And the evolutionary pressure of being hunted produced in 20 million years every fundamental animal body plan that has ever existed. What the Cambrian explosion tells us about life, I think, is something profound about the relationship between adversity and creativity. For 3 billion years, life was comfortable. It fed on light and chemicals. It reproduced by division. It had no enemies in any meaningful sense. And it produced, in 3 billion years, essentially nothing new. And then something started eating it. And in 20 million years, it invented eyes and legs and shells and guts and nervous systems and the vertebrate body plan and everything else. The existential pressure of being hunted, it turns out, is the most productive force in the history of biology. The Cambrian explosion was not life at its most comfortable. It was life at its most threatened. And from that threat came everything. The most recent discoveries, the Jiangchuan biota of April 2026, showing that the roots of the explosion precede the Cambrian boundary, the Huayuan biota of January 2026, showing a second wave of innovation after the Cambrian's first mass extinction, the Saltarella reinterpretation of November 2025, showing the creative diversity of biomineralization strategies. Together paint a picture of the explosion as a process rather than an event. Not a single detonation, but a slow burning fuse followed by a sustained reaction, with multiple waves of innovation and extinction and recovery, all driven by the same fundamental engine: the endless, remorseless, creative pressure of life eating life. That engine has been running for 538 million years. It produced the trilobite and the nautiloid and the anomalocaris and the fish and the frog and the dinosaur and the mammal and the primate, and eventually you. Every tooth in your mouth, every lens in your eye, every bone in your skeleton traces its evolutionary ancestry to the weapons and defenses of the Cambrian arms race. You are, in the most literal biological sense, a product of that war. And the war, as far as anyone can tell, is still going on.